All right, so let's go ahead and get started, everyone. So this lecture is going to be a little different. Historically, this lecture has been one, and as most lecture, is tied around a disease. I'm going to pivot a little bit for this one because I think there's a unique opportunity to do something a little different here. We're going to look at the extracellular matrix in the context of a growing area of research that's tissue engineering and regenerative medicine. I'm biased because that's part of what I do, and so if I geek out a little bit, I apologize. So what are tissue engineering in regenerative medicine? So tissue engineering is fairly new. Funny enough, it was founded by the same guy that founded Moderna, that did the COVID-19 vaccine, Bob Langer up at MIT. So in a study published in 1993 with him and Joseph Vacanti, they wanted to try and figure out for materials that are typically implanted in the body or that have biological relevancy, they wanted to try and figure out if they could use those to meet the growing demand for organs for organ donors. And what they tried to do is try and figure out, okay, if we tried to create some sort of biomaterial that was biologically, usually biomaterials 30 years ago now, that's weird to say, 30 years ago, they typically wanted to reduce how biologically active they were. They wanted to make them bio-inert. But the field tried to pivot here and tried to say, can we make them more bioactive to promote tissue growth and tissue healing? And so what they did was they made a polymer in the shape of an ear, implanted it in the back of a mouse, and tried to see, instead of being bio-inert, can it basically try and create a new ear on the back of a mouse? And so this mouse was what they published in the Study in Science in 1993. It's been dubbed the Vacanti mouse now. And this was the birth of tissue engineering. The field is typically defined as the combination of some sort of scaffold, some sort of polymer material, combined with cells or biologically active molecules, proteins, growth factors, lipids. And the goal of tissue engineering is to assemble, to promote the restoration or improvement of damaged tissues or entire organs. There's a professor at Wake Forest named Tony Atala who has these huge bioreactors where he's just trying to grow bladders, kidneys, livers, and unfortunately, it's turned out to be a lot more complicated, but the field is trying to meet the growing demand for organs, for organ donors. And so to this point so far, the only thing that's really worked clinically is artificial skin or cartilage, because as you can imagine, there are incredible regulatory hurdles for tissue engineering, because for just a scaffold, you have to deal with implant regulations. For just cells, you have to deal with an active biologic regulatory hurdles. And for proteins, lipids, or whatever, you have to deal with drug regulatory hurdles. In tissue engineering, we combine all of them. You have to deal with all three regulatory hurdles. And so the field is trying to figure out how can we promote the vision of what tissue engineering is with trying to create a more simplified and less cumbersome therapy that has all these regulatory hurdles that I just described. Now, that's tissue engineering. What is regenerative medicine? Regenerative medicine is a much broader field, a much broader term that tries to describe research that it includes tissue engineering. It's under the umbrella of regenerative medicine, but it's a much larger field that tries to figure out how to promote healing and also potentially try and leverage the self-healing capacities that we have inherently. Oftentimes, if you have some sort of bone fracture, if it fits within a certain set of restrictions, your bones will naturally heal. And one example of tissue that doesn't typically heal is neural damage. And so in regenerative medicine, outside of tissue engineering, these are approaches that try and promote healing that sometimes otherwise won't happen, or to try and accelerate it. So the terms tissue engineering and regenerative medicine are oftentimes used interchangeably. Like for example, I'm a member of the Tissue Engineering Regenerative Medicine International Society. It can sometimes just be used interchangeably like that, but they actually have very different meanings, because there are people who are involved in regenerative medicine but don't do tissue engineering for some of the same reasons I described earlier, because of all the regulatory hurdles that come along with it. So oftentimes one of the ways that people in tissue engineering or regenerative medicine try and create therapies or their approaches are oftentimes informed by what is present in the body normally, to mimic what's there. And oftentimes they're trying to mimic the extracellular matrix. That's the scaffold part in tissue engineering, is oftentimes trying to replicate that. So the extracellular matrix, to try and provide a broad definition of it, there are proteins and polysaccharides that are locally secreted by cells and assembled into this matrix. If you can envision, when the framing of a house is being built, as the process is happening, or rather maybe a building, and you have like a scaffolding around the house, that scaffolding is kind of like the extracellular matrix in a way, because it's providing at a very base level, kind of like the structural cues for what is taking shape here. And so we have two main classifications of extracellular matrix in biology. One is the basal lamina, or another term that's oftentimes more used today is basement membrane. Another classification is the connective tissue. So today we're going to try and tackle basement membrane versus connective tissue. And as you can appreciate here, we've talked about in the past epithelial cells versus mesenchymal cells. And you can see in the top right image that there's a sheet of epithelial cells that are present. And sorry, I should say the bottom left image. And the yellow is an indication of the basal lamina or the basement membrane. So among sheets of cells, there's typically a basement membrane right underneath it. But then underneath all of that, underneath the basement membrane, is this broad connective tissue where there are cells and fibers and proteoglycans, and polysaccharides that are present that help give shape, provide cues, and are critically important in biology. And so in the bottom right, that's an electron micrograph of what you can see is a sheet of cells on top and a basement membrane right underneath. When you use an electron microscope, the basement membrane looks literally like just a sheet underneath these cells. So what physiological role does the ECM, yes, go ahead. So the basement membrane is going to be right underneath epithelial cells. It'll be right underneath, anytime there's like sheets of cells, you're going to have a basement membrane under there. And you're also going to have cells that are on the, like outside of blood vessels sometimes. And it's oftentimes, it's also kind of a sheet. They're not epithelial cells, but they're also a sheet of cells. And so anytime you have a sheet of cells somewhere, also in the like glomerulus of the kidney, you're going to have endothelial cells and epithelial cells. And you're going to have, they're going to be like, I mean, it's wrapped all up, but they're, a continuous sheet. And you're going to have a basement membrane under each of those. The connective tissue side is going to be basically everywhere else in the body. So like anywhere you have, so the skin is a great example. So, the skin, we have, what's this doing this now? There we go. Skin, we have an epidermis. We have epithelial cells, they're on the surface. Various kinds of cells are also in there. Underneath that is the dermis. You have an ECM there, and you also have connective tissue that has nerves, blood vessels, and lots of different cells that are present. Let me go back one slide, forgive me. Are all present here. So with the basement membrane, you're going to have a lot of cells with a very small, thin basement membrane. In the connective tissue, you're going to have a ton of protein, like a gelatinous protein here, with some cells that come through, some veins, some nerves. And so they serve different functions, but they're also situated differently. That's kind of ties back to their function here. So their physiological roles is structurally and biological. The structural roles is, like I described earlier, is scaffolding. It actually provides a structural cue for what the shape is going to be. It's also going to provide tension. So if you think about I was just describing the skin having an extracellular matrix. If you think about the skin and the tension, if you pull on the skin, it's only going to keep going so far and it's going to stop. But also the way that the skin's really interesting because there's kind of different tension lines in the skin based on how we move. So like if you think about it, I thought this was cool. This might be weird to some, but like you can pull skin this way, but you can't pull skin this way. Like there's these they're called Langer lines. There's like the way skin is, or the way our proteins are situated in our skin informs how it's supposed to move. You're good. It's not an invitation to pinch each other and test each other's Langer lines. In addition to that, there's a cushioning role that the ECM plays, especially in our cartilage. If you think about over years of time, you can think about athletes whose cartilage they wear down because their cartilage is meant to provide that cushioning, and over time that the ECM components actually begin to degrade and to get worn down, and that cushioning is gone, and so it plays an important role there. It also can be a molecular filter, like I described with the kidney and the glomerulus. It actually is between our bloodstream and the urine. It also provides a filtering function there. It can serve as a boundary between different tissue types. One thing that we'll talk about later is that it can actually serve as a storage depot for proteins, growth factors, and cytokines. And it can actually block cryptic sites. We'll also talk about that a little bit later, that there's these hidden parts of proteins that ECM will block, and it can actually, via enzymatic degradation, can then reveal these cryptic sites that have actual biological consequences. The biological roles, cell polarity, that's kind of a directionality of cells. And going back to like the epithelium, there's a top and a bottom. The top is the apical and the basal is the bottom. The basal membrane will only be present on the basal side of the epithelium. And so the cell polarity is a huge area of research, and the basal membrane plays an important role in helping direct that polarity. It provides important cues for cell adhesion to one another or to the surrounding connective tissue. Really important in morphogenesis and differentiation. Morphogenesis is kind of the development of tissue, and differentiation is kind of the specification that cells go through. So when a stem cell is trying to become more specialized, that specialization process is called differentiation. And there was a really cool study that showed back, I think it was in 2010, that if you just change the ECM proteins that a stem cell is on, if there are ECM proteins that are from bone, that stem cell is going to become an osteoblast. If you provide cues that are present on the blood vessels, they're going to become smooth muscle cells that are wrapped around blood vessels. And so like the ECM can actually provide the cues for stem cells to specialize and become whatever the ECM is originated from. ECM also provides cues for migration, for proliferation, and for prevention of apoptosis. Can anyone, by raising your hand, tell me what apoptosis is? Yeah, go ahead. Programmed cell death, exactly. The process of apoptosis is informed by the availability of something for the cells to anchor onto. So when cells are anchored onto their extracellular matrix, just for context, I'm going to keep saying ECM. That's a much easier way to, that's an easier way for you to write it and for me to say it. So I'll be saying ECM a lot in case that's confusing. When a cell is anchored down to its ECM, that is one cue that tells a cell to not die. I have something here. I'm going to pull, tug, and undergo different processes. But if that anchorage is gone, the cells will undergo apoptosis. So the ECM can prevent apoptosis in some ways. You stole my line. There are some cells that are not anchorage dependent. Red blood cells are not they're not nucleated, and so they don't have some of the same processes. But there is another group of cells that are in the bloodstream. Is there another one you can think of? White blood cells, yeah. So white blood cells or immune cells they do not have anchorage-dependent prevention of apoptosis. And so, yeah, so great question, and you stole my line. These tables are not things that I'm expecting to memorize everything, but rather help provide an understanding or a big picture of the roles that the ECM can play. The ECM proteins, whenever they are knocked out in mice, knocked out means that the gene is deleted in a blastocyst that then grows up to be a mouse, and you can, over time, develop mice that completely lack one gene. And oftentimes when we do that in science, we also have to ask the question, is this lethal? Like, can they survive without this single gene? A lot of times they're fine, but there are cases where they will not survive. And if you take a look at that table to the right, a lot of knockout mice for different ECM proteins are lethal. Interestingly enough, when we are 2 cells old, for lack of a better phrase, we already begin to express laminin. And by day 4 in a laminin knockout mouse, by day 4, the blastocyst dies. And so many of these ECM proteins are really important for development and the actual formation and creation of different organs. Yes? So that's an entire field of study. No, that's fine. That's a great question. There's an entire field of study. We had one guy, he just left, Cody Ravichandran. He just got poached to go to Washington University in St. Louis, but the entire study of apoptosis, there's different cues. So like there are some ECM biologists that are studying it from the perspective of, can we provide really careful cues in the ECM that might trigger apoptosis? There are other people that are examining it from the perspective of immune cells that, there are a few key proteins and receptors that immune cells can provide to cells nearby to induce apoptosis, but it's not just a one size fits all. This is how it happens. Does that help?
Yeah.
Okay. Yeah. So we looked at the ECM as 2 classifications of basal membrane and connective tissue. Now we're going to kind of dive a little bit deeper. So the main biomolecules in the ECM are fibrous proteins that are usually referred to as glycoproteins, and another are glycosaminoglycans. Again, for your shorthand notation and for my own breath, I'm going to say gags. That's typically how we refer to them. Glycosaminoglycans, as the name implies, it's oftentimes a repeating disaccharide unit. So saccharide is oftentimes longhand for sugars. So it's this repeating sugar unit of 2 sugars, 2 sugars, 2 sugars, 2 sugars, 2 sugars. And sometimes those gags, those disaccharide units, can be bound to a core protein. And when that happens, that's referred to as a proteoglycan. So to try and alleviate confusion, the shorthand trick that I derived over time to try and alleviate confusion from proteoglycans to glycoproteins to gags. Proteoglycans, so the phrase here, so the suffix, the glycans here, whatever is the suffix indicates what there's the most of. So with proteoglycans, it's mostly sugar. And that's illustrated to the bottom right here. And glycoproteins, proteins is a suffix, it has mostly proteins. So it's a protein that has a couple of sugar moieties attached to the polypeptide chain. And so we have in the ECM, as illustrated below to the bottom left, we have different kinds of proteins. The proteins are denoted in green, and the gags are denoted in purple. I think that's purple. I apologize if I'm wrong to anyone listening to this recording. So we have laminin, type 4 collagen. Those are some key basement membrane proteins that we're going to talk about more in a moment. But we also have to the right perlikin, which is a proteoglycan. because it has a small core protein and a lot of a long set of gag chains to the right of it. We have hyaluronin, which is exclusively a glycosaminoglycan. It is one of the largest gags that we have. It's just a long chain of disaccharide units that are repeating. And additionally, below we have decarin and aggrecan. Aggrecan is enormous. So we talked about before that proteins are typically 50 kilodaltons. Aggrecan is, I believe, 3 megadaltons. And so it is a giant proteoglycan with hundreds of repeating gag units tethered to the core protein. So the basement membrane typically has a few different sites where it's, yes. So that's a good question. So the dots are the protein unit. So perlikin is a proteoglycan, and so this is the core protein. So every proteoglycan has a small core protein, and typically a lot of saccharide units attached to it. In this case, perlikin's smaller, but it has a core nuclear protein with gag units attached to it. So it is, in this case, because it has both, because it has mostly saccharide units attached to it, this makes it a proteoglycan. If it was, so like for these, we're showing them as only proteins, but in actual biology, they have small sugar units attached to them that oftentimes can dictate their biological activity. And so these are glycoproteins, even though we just show them like a simple illustration that shows them as just a peptide, a protein fiber. Are there any other questions? All right. So the basement membrane typically is located in 3 different locations in our bodies. A couple of them I talked about earlier. One is that they underlie the epithelium. And again, they lie on the basal side, the underneath side of an epithelial sheet or epithelial monolayer. Another example is that they can also surround cells. So with individual muscle fibers, they will have their own basement membrane wrapped around them. And the last one that I mentioned earlier is that they can be in between two sheets of cells. That's hard to say fast. 2 sheets of cells. And an example of that is the glomerulus in the kidney, that there's a basement membrane that serves as a filtration function between the endothelium, the endothelial cells, and the epithelium in the kidney. So the basement membrane has 4 main components. Their 2 main proteins or glycoproteins are laminin and collagen 4. And the 2 other components are nitrogen and perlikin. Perlikin and nitrogen both serve a cross-linking function. So one thing I'm going to repeat, make sure I want to try and repeat several times, is that the basement membrane, as it's being assembled, you have laminin that is assembling and polymerizing together. So you'll have a sheet of laminin, and then you'll have collagen 4 that's being expressed, released, and they're polymerizing together. But so you have 2 different sheets of proteins, of glycoproteins. Nitrogen and perlikin will actually bridge and cross-link them together. And so the basement membrane is an incredibly thin sheet. It's 40 to 120 nanometers thick, but it's two polymer networks that are cross-linked and bridged together. And that bridging or cross-linking is accomplished by nitrogen and perlikin. So we're going to take a quick look at each of the components. So laminin is a heterotrimer. Does anyone want to take a moment to describe what a heterotrimer, what that means, that it's a heterotrimer? Exactly right. It is 3 different subunits that are together. We have alpha chains, beta chains, and gamma chains. So I remember we talked about for a couple lectures now that chains mean nothing. That's typically for light chain and heavy chain. The light and heavy denotes that it's, they were run on a gel and enzymatically degraded, and seeing that there's a distinction there. Now, when we're talking about multiple peptides coming together to form a protein complex, this is where chain can mean something different. So there are genes that promote the expression of laminin alpha chains. There are genes that express laminin beta chains and laminin gamma chains. And so what will happen is that they're expressed, they'll come together in the Golgi apparatus, they're assembled together there, and they're released. And so because of the diversity we have with laminin as a heterotrimer, there are 16 different isoforms that have been identified. And the way they're denoted typically is the number, each different chain has a number associated with it: alpha 1, beta 1, gamma 1, alpha 2, beta 2, gamma 2. And so each laminin chain is denoted by whatever number the alpha, beta, gamma is. So laminin 111 is a laminin that has the alpha 1 chain, beta 1 chain, gamma 1 chain. And it is, as I mentioned earlier, it's a key part in the basement membrane. There's, as you can imagine, because it's two different sheets, laminin constitutes about 50% of the protein content by weight of the basement membrane. And each different chain or each different part of laminin has a different interaction or a different function. So laminin historically has been dubbed as like the biology's cross, or like the God protein. And so the short arms of laminin are the beta and gamma chains, and then the long arm is the alpha chain that comes down horizontally on the bottom left image that you have here. And it's been identified in the past based on methods called rotary shadowing. That's what's to the bottom right. You can actually historically image like single molecules and get a sense of like what their actual structure is. Now we have even better means of doing this. So like I suspect that image is from decades ago. We have something called cryo electron microscopy, where we can actually like have almost like 100x zoom of what's there in the rotary shadowing image. And so sometimes what is considered what we have a consensus about is actually sometimes challenged by newer methods. As an example, we're probably not going to get to it, but in the lab I'm in, we study one ECM protein called fibronectin. Everyone believes its structure. We know it. It's been imaged by rotary shadowing. We just discovered something that maybe tells us like, oh, we might have the structure completely wrong of fibronectin and what it looks like at the secondary structural level. So that's a great way to say maybe half of this stuff is right. Who knows? But with that being said, that's a great way to motivate you to tune in, I imagine. Yes, I'm sorry, go ahead. How the laminins what, I'm sorry? So we have alpha chain, beta chain, and gamma chain. So the 111, so it's first the alpha, first then the beta, then the gamma. So the 111 is an alpha 1, beta 1, gamma 1 laminin molecule. So for example, here, if it was, so for this would be laminin 3,3, because it's the three, alpha 3, beta 3, gamma 2. So it'd be 3,3,2. And so the nomenclature for naming laminin is just based on what kind of alpha chains, beta chains, and gamma chains are present. And you can tell that it also, the different kinds of protein subunits that are present on each different species of laminin can have a different structure. And it's also important to note that each arm has a different function that we'll talk about here. So the beta chain and gamma chains are typically where laminins will polymerize together. They'll grab other laminin molecules and create that mesh of laminin, that laminin sheet. The alpha chain, interestingly, has a more specific role. It'll actually bind to what we call integrins. Integrins are the surface receptors on cells that bind to the ECM. And so for cells that have a basement membrane, one of the locations they'll bind is on the alpha chain, either on the N-terminus or the C-terminus. But what the important key takeaway here is that the beta chain and the gamma chain is where you have polymerization between laminin molecules, and the alpha chain, at the ends of the alpha chain, is where you'll see integrins from cells binding to laminin. The other component of the basement membrane is collagen type 4. Laminin accounts for about 50%, and collagen 4 accounts for approximately 50% of the protein mass in the basement membrane. So while laminin was a heterotrimer, collagen, this is true for all collagens, collagen consists of 3 alpha chains coming together. And so when they come together, they form this helical structure. And if you can imagine, this wrapped helical structure provides a lot of strength to collagen. Collagen provide that structure. One of the things this is true not only just in biology but also general in engineering. Oftentimes structure informs function, and so with collagen, it provides strength here. And the defining feature for a collagen peptide chain is the gly-XY repeats. Glycine, some amino acid, and that repeating throughout the entire collagen protein. Typically, the XY repeats are a key member of that is proline. Does anyone remember a defining feature of proline? Yeah. It's kink-inducing. So what ends up happening if throughout the entire collagen protein, you have glycine, proline, oftentimes the Y is hydroxyproline. We'll talk about that later, just put that in your back pocket. The proline will end up kinking, and that's what promotes the wrapping of the collagen alpha chains and produces this triple helical structure. So what's different with collagen type 4 is that there are typically less prolines in the XY. So that typically means that there are less kinks and it makes it more flexible, which is helpful because collagen type 4, the basement membrane, it's a sheet. It kind of sits underneath cells. Whereas if you think back to that image that I had a few slides ago, where we had an epithelial layer, the basement membrane, and then like connective tissue here, like this like gamesh of cells and fibers. The ability for, so I guess maybe it'll help to go back to the skin. So, the flexibility underneath the epithelium is pretty important, but everything underneath that will want to provide tension. And so the less rigidity in collagen type 4 is important for its role as a member of the basement membrane. But other types of collagen, right now I think we're sitting at 29 types. Other types of collagen will oftentimes have a less flexible structure because of what they need to be able to do. For those that are in the bone, it's really important for them to provide strength. In the blood vessels, in the lung, they need to be able to withstand different pressures and provide strength and rigidity. For the basement membrane, collagen type 4 has less proline so that it can provide a little more flexibility as a basement membrane. Yes?
Definitely a dumb question.
I love dumb questions.
But are the alpha chains and laminin, do they how they bind to integrins? Is it the same here with the 3 alpha chains?
Yeah, so there's the alpha chains in collagen are not the same as the alpha chains in laminin. The collagen is among one of the best ECM proteins at promoting cell binding or cell adhesion. There are specific integrins that recognize laminin, specific integrins that recognize collagen, and other integrins that recognize other stuff. But the fact that this has only alpha chains and not alpha chains on laminin or bind integrins, it's not the same. They're totally different genes and totally different proteins. Was not a dumb question. That was a great question. So nitrogen is a glycoprotein, and it's again, it's one of the components of the basement membrane that helps to bridge and cross-link the laminin sheet with the collagen type 4 sheet. And it is, there's typically two isoforms, and it's a glycoprotein. But again, the key takeaway from this is that it helps bridge the two different the two different protein sheets in the basement membrane. And the other member, the last member of the basement membrane is perlikin. It's a proteoglycan, so that means glycan is the suffix, that it's mostly sugars. And so this is, the sugars that are on it are heparin sulfate. Oftentimes with proteoglycans, sometimes on the front end, they'll kind of identify the species of, or the kinds of gags that bind with they're part of the protein unit. So perlikin, the predominant species of gags is heparin sulfate. And it helps bridge laminin to collagen type 4 in the basement membrane. Are there any questions there? All this stuff is exciting. I think connective tissue is more interesting, but basement membrane is still pretty cool. But we'll go ahead and move on. So how is the basement membrane assembled? The basement membrane proteins are typically assembled in the Golgi complex or Golgi apparatus. What typically happens is when these proteins are expressed, they're each individual peptide chains. So laminin has the alpha, beta, and gamma subunits. The alpha subunit is a polypeptide chain, just like a linear chain. You'll have 3 linear chains that are transported to the Golgi apparatus, and it's in the Golgi where they're then assembled. But what's really important is that for both laminin and collagen type 4, when they're being assembled in their pro-protein form. Does anyone remember what pro-protein form means? Yes. Yeah. And so it's the precursor form. And can anyone venture a guess why that's important? No, with the knowledge that these these proteins will want to polymerize together and assemble, why is it important that they're formed first in their pro-protein form? Yeah. Yeah. If that's exactly right. You don't want it to be assembling and just fill and rupture the Golgi. There are studies that people did where they removed the amino acids for the propeptide of collagen. And what ends up happening is that when you do that, it is exactly like he said, they're gonna start polymerizing, filling up the Golgi, filling up the cell, and just basically destroying the cell. And so the ability to, the presence of that propeptide, I like to refer to it sometimes as a cap. So that pro cap is really important so that when it's shuttled out of the cell, it's actually a pretty, I think this is an example where biology can be really elegant at times. That feels really nerdy to say out loud. That's an inside thought. But so in the Golgi, it's assembled. It has a propeptide cap. What's interesting is that on the surface of cells, oftentimes, they have bound to their surface enzymes that as collagen and laminin are being transported out, they'll clip the propeptide caps off. And collagen, and they'll actually, other integrins will also grab onto laminin, assemble it, and let them polymerize together. The integrins for collagen will grab collagen, assemble them into a layer. Nitrogen and perlikin will cross-link them. And so the cells, the epithelium, or cells that have a basement membrane underneath them, they're creating their own basement membrane, reorganizing it, assembling it, modulating it. And so the important thing to remember here is that we have the propeptide cap so that they won't assemble inside the cell. They're transported out. We have enzymes on the surface that'll clip off the caps, the propeptide caps. And the cell itself will then assemble them together and help physically grab them and move them together. They'll polymerize together, and you'll have a basement membrane right there underneath the cell. And as I mentioned, the nitrogen and perlikin will help bridge and cross-link those two, the laminin sheet and the collagen type 4 sheet, so that you have a copolymer, two different polymer sheets together, creating this basement membrane. Another really, I think, pretty fascinating example of the way biology can be really elegant is that, and this is leveraged in tissue engineering, is that these basement membrane proteins can provide instructive cues for different biological activities for cells. So this is the C terminus of laminin, of the alpha chain. And these little protein globs here are the laminin globular modules, the LG modules. And so different modules will be bound and recognized by different integrins. And so integrins have an alpha and a beta head, and there's a ton of different species for each, and you'll get 24 different integrins usually. And we refer to integrins. Remember, integrins are the receptors that bind to the extracellular matrix. In this orientation for laminin, it'll typically bind to what's called alpha 6 beta 1 integrin. And what's really fascinating is that when alpha 6 beta 1 recognizes laminin in this conformation, it will promote migration. So if you think about you're chopping vegetables for dinner, you cut your hand. What's happening at the cellular molecular level is that alpha 6 beta 1 from local cells are binding and recognizing laminin in this conformation with the 1, 2, 3, 4, 5 LG modules. That's promoting migration. These cells are moving and want to move and fill the gap, bridge the gap of that cut. And then what's going to happen is that there's an enzyme called plasmin that'll cut off a couple of these LG modules. And it's going to expose, cryptic usually indicates like a hidden site that is then available later on based on either enzymatic cleavage or, and I think we won't get to this today, but sometimes cryptic sites can be revealed just by these proteins being unfolded and actually like physically pulled open. This cryptic site will bind to a integrin now, the alpha 6 beta 4, and that's going to induce cell-to-cell adhesion. So here, what's happening is that these cells are migrating across the cut. And here now, after plasmin is cleaved off these LG modules, it's going to bind to alpha 6 beta 4, and it's going to actually promote epithelial cells to bind to each other to fill that gap. And so this is an example of how different sites that can become available on ECM proteins happen typically in healing. And when I first cut my teeth on research, we tried to take certain peptides from that site, put it in a polymer, and see if we could do the same things. Didn't work that great, but it was still fun. And so these can also provide cues for therapeutic approaches, trying to leverage the different cryptic sites that provide different instructive cues. All right, connective tissue. Are there any, before we jump to connective tissue, are there any questions about what we covered for the basement membrane? Mm-hmm. What stops the basement membrane from being built up too much? So typically, the presence, so there's a few different, biology usually has a few different fail-safes. So if there is a need for more basement membrane, the epithelium or other cells will create that basement membrane. And once there is sufficient physical cues that the basement membrane is there, as well as now like chemical cues from like the presence of now type 4 collagen laminin, it's going to provide a feedback loop to say, okay, now we can shut this process off and stop laying down more basement membrane. All right, time for the connective tissue. This is where the fun begins, guys. I know you guys are on the edge of your seats, so I want to keep you posted on the exciting stuff. So typically the connective tissue ECM, it will sit beneath the basement membrane. So the image to the top right, we have the basement membrane underneath the epithelium. And everything under that is the connective tissue ECM. And so it is a site where there are typically more ECM protein. So if you think about the basement membrane versus connective tissue, typically, I think I'm getting ahead of myself here. The epithelium in the basement membrane is really thin. There's a ton of cells there with a really thin, tiny amount of ECM protein. In the connective tissue, it's opposite. You have a ton of ECM protein with sometimes some cells littered throughout it. Sometimes you might not have any cells. But it's this like interstitial space underneath the epithelium where you'll have immune cell. It's this unique space where you'll have fibroblasts, immune cells, blood vessels, nerves, bone. It is an incredibly diverse location where sometimes the ECM, which used to be thought of as just like either structural or adhesion, or sometimes during morphogenesis or development, promoting the development of tissue. But now in tissue engineering or regenerative medicine, we're realizing that it's kind of coordinating a lot of interactions between all of these different cells here. Got really excited and I lost track of myself. Okay, so the components. You have protein fibers that are embedded in an amorphous mixture. So this is typically kind of almost like a gel-like, it almost has gel-like properties because of the proteoglycans. Proteoglycans, with the gags that are associated with them, are very hydrophilic. They love water. They want water. And so they're typically, and that feature is really important, is going to become really important in cartilage and helping to provide the shock absorbing function of cartilage. And the connective tissue ECM, if you think about bone or blood vessels or lungs, it provides the physical cues, or it determines the physical properties. So the ability of our lungs to expand and retract, the ability of our blood vessels to have a pulsatory movement in response to each heartbeat, the ability for it to go out and come back, that strength is imbued by the connective tissue ECM there. So the cell that's near and dear to my heart is the fibroblast. It is the predominant cell type that will secrete and arrange the connective tissue ECM. And it's going to secrete the fibrous proteins as well as the gags and proteoglycans. And typically it's part of what will help pull together, like going back to the example of chopping vegetables and cutting your finger. It's because of its, going back a couple lectures now, because of its actin cytoskeleton that can polymerize and promote these really strong stress fibers. Their integrins can grab one side, their integrins can grab the other and pull together and help retract an injury site, and the epithelium will come and grow back in. Now, what do you think happens if if fibroblasts are producing too much collagen and they're pulling on things too much? What do you think can happen to that wound? Go for it. Exactly, you get a scar. So what typically happens is, if you think about it, if you think back to any large scar that you have, that's typically a response from too much collagen being created and typically too much contraction going on by the fibroblasts. And so we unfortunately fibroblasts in adults are creating scar tissue, whether it's in the skin after a myocardial infarction. I study pulmonary fibrosis, whether it's pulmonary fibrosis. Even after COVID-19, everyone that has long COVID has really severe lung fibrosis. And it's a matter of trying to figure out how can we control and regulate these fibroblasts. And yeah, I'll stop there. I could keep talking. Yeah, so the key takeaway on this slide for you guys is that fibroblasts are secreting and arranging the ECM, and they're expressing, or they're secreting both the fibrous proteins and the gags and proteoglycans here. So now we're going to look at each component of the connective tissue ECM. So gags, the glycosaminoglycans, like I mentioned earlier, they are repeating disaccharides, repeating 2 sugar units. And as an example, we have 2 different kinds of gags here. We have hyaluronin. You'll sometimes see hyaluronin also written as hyaluronic acid, but they're functionally equivalent. And so you'll typically have an amino sugar or a uronic sugar, but the type of gag is oftentimes the specificity of it is distinguished by what type of sugars are there, what type of linkages are there. So if you look at hyaluronin versus heparin sulfate here, the linkage between the sugars is at different carbon locations compared to heparin sulfate here. And so the types of sugars, the way these sugars are linked together, and also for stepping out at looking at the entire gag molecule, the number and location of sulfate groups that are on there. Most gags will have a certain degree of sulfate groups added on. There's typically only one that doesn't have any sulfate groups, but I think we'll get to that maybe. But we have 4 main groups of gags. We have hyaluronin, chondroitin sulfate, heparin sulfate, and keratin sulfate. As you can imagine from the name, keratin sulfate is oftentimes found in the hair and nails because we have a lot of keratin there. Chondroitin sulfate is really interesting. We also find it in the brain as well. And the scars that form in the brain are going to be chondroitin sulfate rich locations. And so like, again, kind of going back to scar formation as being like uncontrolled ECM deposition, in the brain, it's going to look like chondroitin sulfate, whereas a lot of other places, it's going to look like a collagen-rich, oftentimes, it's a reductive way to say it, but a really collagen-rich scar. So these gags typically have really large and extended conformations, and this goes back to their hydrophilic nature. Because our body is so, they will want to expand and be as linear as possible. Like you can imagine if you're like riding on the bus around grounds, like the really obnoxious person pre-COVID, I guess, really obnoxious person that's like yawning, like pushing someone around, like that's what gags want to do. Like they want to expand and be as linear as possible within whatever confined space that they have. And so because of that, they have a high volume to mass ratio. Relative to their mass, they want to expand and take up as much volume as possible because they're so hydrophilic in an aqueous environment. And because of that, their ability to expand like that creates these like interstitial interstitial pores in the ECM that can provide a space for cells to migrate through. So, typically, we have lots of different fibrous proteins in the gags in our connective tissue ECM, and the ability for these gags to push things aside, to want to stretch out as much as possible, creates space for things to bind to our ECM proteins like growth factors, but also provide space for cells to push and pull through. The negative charge that gags have will often attract cations, and the hydrophilic nature of gags and their ability to push on things around them is informed by the high osmotic pressure, and because of that, it permits them to provide high resistance to compressive forces because of that. It is 3:15, so I'm going to call it right there. But thanks for giving me your time, and you guys have a good day.